Hello, students. So today we will be talking about chapter 7, membrane structure, and I'm going to share the PowerPoint slides with you so that we can explain what exactly is membrane structure and function. As the title suggests, we are looking at our plasma membrane structure and what are the number of functions that our plasma membrane is performing in our bodies. So our plasma membrane is said to be either selectively permeable or differentially permeable or semi-permeable, which means that it's going to select or choose small substances in the form of ions or gases to move across it. We have different types of proteins like transport proteins, carrier mediated proteins. From the very term transport proteins are proteins that are responsible for the passage of these small ions or gases to go across it. We have phospholipids, which is a class of lipids and fats. And phospholipids consisted of two fatty acids with one glycerol molecule, to which a phosphate group was attached. They are referred to as amphipathic molecules. What are amphipathic molecules? Amphipathic molecules are molecules that have both a head and a tail. And the phospholipid molecule consists of a water-loving head, termed hydrophilic head, and a water-fearing tail, which is called a hydrophobic tail. And these hydrophobic tails of the phospholipid bilayer are present within the membrane. Why? Because they are afraid of water, while the heads are facing outward towards the side of the water that surrounds our cell, because they love water. So we can see clearly in the slide, this is the lipid bilayer. Our cell membranes consist of two phospholipids. So we also refer to it as a phospholipid bilayer. The head is facing the watery environment, and therefore it's termed hydrophilic head. The tails are afraid of water. They are water-fearing molecules. Therefore, they face inward. These are the two fatty acid tails with the glycerol and the phosphate group attached. Makes it a phospholipid. Now, our phospholipid bilayer, which our cell membranes are composed of, is also referred to as a fluid mosaic model. As you can see from the diagram, that our cell membranes are very intricate. They're so detailed. They have a number of structures that are present within it. You have a variety of proteins. These proteins here, which are towards the boundary of the bilayer, are called peripheral. Periphery comes from the word boundary. So peripheral proteins are always towards the cytoplasmic side of the plasma membrane, and they are towards the hydrophilic heads, peripheral. Then you have the other proteins that are embedded within the bilayer, and these are called integral proteins, or also referred to as integrins, integrins. Now you have the outside of the cell membrane and the inside of the cell membrane. This area here, which is outside the lipid bilayer, is called the ECM. extracellular matrix. A matrix is a network of all these fibers. If you are referring to the fluid that is present in the outside the cell, it would be termed ECF, extracellular fluid. This is the cytoplasmic side of your plasma membrane. So now what we see is within the bilayer, you see cholesterol. This is the good cholesterol that our cell membranes are making or producing. On top of one of the sides of the lipid bilayer, we are seeing glycoproteins. Glyco comes from the word glycogen, which is a sugar. In the animal cells, sugar is stored for long-term purposes as glycogen molecules. So you strictly can see, you can clearly see here, these Y-shaped structures are referred to as carbohydrate chains. So when a carbohydrate attaches itself to a protein, this whole molecule that is formed is called a glycoprotein. So you have seen, we see glycoproteins on one side of our plasma membrane. If you look here, when you see only one chain attached, it would be referred to as a glycolipid. So this is a glycolipid. Don't forget, Y-shaped chain, glycoprotein, because that's a carbohydrate chain. If you have one chain, then that's a lipid chain attached to the glyco, forming glycolipids. So we can clearly see the ECM here, the extracellular side of the membrane, and the cytoplasmic side. What do we see on the cytoplasmic side? We see a number of these collagen proteins. These are collagen proteins. So you have a number of proteins that are present within the plasma membrane. Now we are looking to see what would be the state of a membrane. Depending on the state of the membrane would be fluid, and therefore it will permit substances to move across it. So membranes that are held together mainly by weak hydrophobic interactions, and most of these lipids and some proteins, they can move laterally. So when you look at the plasma membrane structure, substances can move laterally. So cholesterol, which is present within the bilayer, which is being made by the cell membrane, what is the function of cholesterol? It gives flexibility to the plasma membrane, to the cells, as well as it allows the substances to move laterally, sideways. So why do we refer to the plasma membrane as a fluid mosaic model? Because it is a mosaic. It consists of a variety of all these protein molecules that are present within the phospholipid bilayer. Therefore, it is a mosaic model, fluid mosaic model. And this model was put forth by Singer and Nicholson. And these proteins that we are seeing within the whole lipid bilayer, they are not randomly distributed in the membrane. Now, what happens to the plasma membrane as the temperatures are going to cool? When the temperatures cool, the plasma membrane will switch from a fluid state to a solid state. And the temperatures at which the membrane solidifies will depend on the type of lipids that are present. So now here we have two different types of cells, a mouse cell and you have a human cell. For the purpose of comprehension, they have put different colors here. But these are the proteins, membrane proteins that are present on the mouse cell and on the human cell. So when the mouse cell and human cell merge together, mix together, they form a hybrid cell. But what do we see in this hybrid cell? this hybrid cell consists of both the membrane proteins from the mouse cell and the human cell, which have been uniformly distributed in this hybrid cell. So it's important to see that there is intermixing of these membrane proteins from the mouse cell and from the human cell, which is evenly or uniformly distributed on the surface. So you can see once these proteins from the hybrid cell are now mixed after an hour, you see that the hybrid cell will now have a combination of both mouse proteins and human cell proteins. So membranes that are rich in unsaturated fatty acids, they are more fluid than the rich saturated fatty acids. Why? We've seen in the macromolecules, lipids and fats, that a saturated fat is solid at room temperature. Unsaturated fat is liquid at room temperature. So the same concept applies here. If the membrane is rich in unsaturated fatty acids, it would be more fluid, more liquid than a saturated fatty acid. And therefore, for membranes to work properly, they must be fluid. And they're comparing the fluidity to that as of salad oil. Salad oil is very fluid. It's not viscous. It's not thick. So we refer to the steroid cholesterol that is being produced within the cell membrane, giving the lateral movement of other substances, making the bilayer strong, giving it flexibility. And therefore, cholesterol has different effects on the membrane fluidity at different temperatures. Now, our body temperature is 37 degrees Celsius. So as our body's temperature warms to 37 degrees Celsius, what happens is cholesterol is going to restrain the phospholipid movement. Once the temperatures drop at cool temperatures, it is going to maintain the fluidity by preventing the tight packing of the phospholipid tails. So though cholesterol is present also, when we think of cholesterol, we never associate with cholesterol in plants, but though cholesterol is present in plants, they usually are related to the steroid lipids in order to buffer the whole membrane fluidity. And so we can see we're comparing unsaturated fatty acids with the saturated fatty acids. And we can see in the phospholipid tails, what are we seeing? Unsaturated tails prevent the tight packing. And because the tails, there is spaces between the tails, it prevents the tight packing of tail. Therefore, it makes the bilayer more fluid, allows substances to move laterally. But in saturated fats, which are solid at room temperature, the oils are very thick, they're viscous, they're dense, and therefore there is tight packing of the phospholipid tails, causing the prevention of the movement of molecules. It does not allow the molecules to move easily. So we can see that cholesterol reduces membrane fluidity at moderate temperatures, but at low temperatures, it is going to hinder the solidification. It's an important concept. You will get a question, or you can get a question on the fluidity and the viscosity of the plasma membrane. We looked at peripheral proteins. I told you what peripheral proteins were, what integral proteins are. Now, in addition, the cell surface membranes are going to carry out several functions. They're going to perform transport, enzyme activity, signal transduction, cell recognition, intercellular joining, and attachment to the cytoskeleton and the ECM, which is extracellular matrix. So we can see here the N-terminus side, and we can see the C-terminus side. The alpha helix we saw, which was the protein structure, the beta protein structure, secondary structural protein, which was the alpha helix and the beta pleated structures. And so it had the two sides, N and the C terminus, And we can clearly see this, the C terminus would be the cytoplasmic side of the molecule. So we're going to see what are these membrane proteins functions here. They're carrying out, they're conducting transport. They're transporting substances, but what substances can move across the cell membrane? Small molecules or substances in the form of ions and gases. And so we can see if normally all substances are going to move from an area of high concentration to an area of low concentration, that's passive transport. And therefore, if they move from high to low, no energy is required. But if they move from low to high, see, I had to push my hand up because they're going from low concentration to high, then energy is required by the cell to move or transport the substances. What is the energy currency of the cell? ATP. And so we can see here that ATP is being used to shuffle out the larger molecules across the cell membrane. Let's look at enzymatic activity. We know proteins' function is they act as enzymes. Enzymes act as catalysts. And we know catalysts are chemical substances that accelerate or speed up a reaction. Now, for enzyme activity to take place, we need what we call a substrate. But the shape of the substrate and the enzyme has to be the same for that substrate to go and bind to the enzyme. Now we can see different shapes here of the molecules. You have a circle, you have a square, and you have two opposite triangles like a bow-shaped structure. Now, which of these molecules can fit here? If this is your enzyme and these are your substrates, so these substrate molecules, this one will be able to go and fit here because of the M-shaped structure. This cannot fit here and this circle cannot fit here. So, and vice versa, these can't fit into each other because remember, shape determines function. And so this will go, this substrate can go and fit here. And when the enzyme goes and fits into the substrate, it forms what we call an enzyme substrate complex. So that is how enzymes are performing its function in the plasma membrane. In signal transduction, transduction is to relay the signal. So here you have the receptor. We have a number of receptor molecules on our cell membrane, and a receptor from the very term is going to receive a signal. And in signal transduction, what is happening, you can see the signaling molecule here is going to bind to the receptor molecule for the transduction to take place. Then you have cell-to-cell recognition. And we saw when you see a white chain, you know immediately it was a carbohydrate chain attaching to a protein, forming the glycoprotein. So the cell is recognizing that this is a glycoprotein, this is a glycolipid, and it will allow the molecule to move across the cell membrane. So this is important in understanding what is going on within the cell. Another function that the cell membrane is going to perform is intercellular joining. You see, intercellular meaning between the cells there is a space. And so substances can move across the cell membrane there. And last but not the least is attachment to the cytoskeleton and extracellular matrix. So we can see these are fibrinogen molecules, and these fibrinogens, which are different types of proteins, etc., how they are attaching themselves but to the cytoskeleton, but on the ECM, extracellular matrix side of it. So these cell surface marker proteins, they are extremely important in medical field. And we have the HIV. You have proteins that are cell surface proteins that are present, and the HIV must bind to the immune cell surface protein CD4, and it has to have a co-receptor CCR5 in order to infect a cell. HIV cannot enter a cell of resistant individuals who do not have that CCR5. So for an individual to suffer from HIV, they have to have the co-receptor CCR5 to go and bind to the surface protein called the CD4 protein in order to cause the infections. And now drugs are being developed in order to mask that CCR5 protein, which is the co-receptor that infects a cell. So cells can often recognize each other by binding to molecules. They often are going to contain the carbohydrates on the extracellular matrix part of it. And now this is the illustration of HIV can infect a cell with CCR5 on its surface, as in most people, but HIV cannot infect a cell. So for example, I don't suffer from HIV. Why don't I suffer from HIV? It's because I do not have this co-receptor. So you have a receptor and a co-receptor molecule. The co-receptor has to go and bind to the HIV to cause the infection. In this case, the receptors are only CD4. Everyone has these receptor molecules, the CD4 receptors. But if you do not have the co-receptor CCR5, which needs to go and bind to the receptor molecule in order to infect the individual and cause HIV. Okay. So we had the transport proteins, which we talked about earlier, which are allowing the passage of hydrophilic substances to move across the membranes. So it's specific for particular substances that it is going to move. But some types of transport proteins are called channel proteins. And these channel proteins, from the very word, they're acting as pathways, channels. So they have a hydrophilic channel that is going to shuffle small molecules or ions to move across it. It acts as a pathway, as a tunnel. But channel proteins, a specific type of channel protein called aqua protein. Aqua comes from the Latin word, which means water. So aqua proteins are specifically going to only facilitate the passage of water molecules across the cell membrane. So do not forget that aquaporins is a type of channel protein, and the type of channel protein and channel proteins are one type of transport proteins. We have other types of transport proteins, which are called carrier proteins. Think of the word carrier. To carry us to move across. So carrier proteins are going to bind to the molecules, and they're going to change the shape of the molecules, thereby enabling that molecule to move across the cell membrane. So now we come to, as I mentioned earlier, there are two main types of transport: active transport, passive transport. Now, when the analogy I use is, what am I doing? Am I being active or passive? And I am being very active because I'm explaining, I'm talking, so I'm using a lot of ATP, the currency, the energy of the cell. So active transport, you have two types: endocytosis, exocytosis, which I'll talk about a little later. But if you are just sitting and listening to my recordings, you are passively listening. You are not using energy, much energy. Therefore, passive transport is the process where substances are going to move from an area of high concentration to an area of low concentration, along or down a gradient with no energy required for the movement. So the three keywords in passive transport are high concentration to low concentration, along or down a gradient, no energy required for the movement. Passive transport is of three types: diffusion, osmosis, facilitated diffusion. So you will be working on conducting experiments on osmosis and diffusion in the lab. And so it will bring more clarity to the theory that I'm explaining. So let's understand then, what is diffusion? What is the meaning of to diffuse? To diffuse is to spread. So for example, if I took a beaker and I added salt, my solute, to the beaker, what would happen? You stir it and you would see the salt dissolving in the beaker until a point is reached where there is no more salt, because now you have a solution which is salty water. Or let's take a more vivid example. So if you took chocolate syrup, or if you were making a juice, Tang crystals or Kool-Aid crystals, and you dropped, say, Kool-Aid blue crystals into your water and you were making a drink, you would see you've dropped it from high concentration to low. And so what would happen is, after some time, you would see the whole cup of water turning completely blue if you added blue Kool-Aid crystals, showing you that the crystals move from an area of high concentration to an area of low concentration until all the molecules, all the crystals were evenly distributed. And so your whole cup now has turned blue and you're going to drink the Kool-Aid. So that's the process of diffusion. So you could have, I just explained diffusion of a solid. If you took syrup, chocolate syrup, that would be diffusion of a liquid. And gas would be, if you boil water, what happens to the water? It starts bubbling. Gaseous vapor is, or condensation is occurring. So diffusion of a solid, liquid. Another classic example of gas. If you keep spraying your house with air freshener, hairspray, or perfume, you see the gaseous molecules moving from an area where you sprayed it, and then slowly from high concentration, they move to another part of the room. And by the time the gaseous molecules or the perfume or the air freshener moves to the rest of the room, you start getting the whiff of the odor. Somebody enters into the room from another room and says, wow, I'm getting nice perfume smell. It's because of the gaseous movement of the molecules. So we've talked about all three scenarios: diffusion of a solid, liquid, and a gas. And that's one example, one type of passive transport. So let's go to the second one, which is osmosis. What is osmosis? Keyword: osmosis is one type of diffusion in which water molecules move across a semi-permeable membrane. No other molecules can move. It's only water that is moving across the semi-permeable cell membrane. And of course, the water molecules are moving from high concentration to low concentration. So until equilibrium is maintained or reached. So we have a term called tonicity. What does tonicity come from? It is the ability of a surrounding solution to cause the cell either to gain water or to lose water. And therefore, tonicity is going to depend on the concentration of the solute that cannot move across that cell membrane relative to what is inside the cell. So we have three different types of solutions, extremely important. We have isotonic, hypotonic, and hypertonic. Iso, I'm going to go to the illustration here so that we can understand it better. So these are the three different types of solutions: isotonic, hypotonic, hypertonic. I'm going to give you the Greek derivatives of each of these types of solutions. We see this is exactly happening to the red blood cells within our bodies, the RBCs. Now, what then is an isotonic solution? Iso comes from the Greek word, it's derived from the Greek word isos, which means same or equal. So when the concentration of solute outside the cell, the concentration of solute outside the cell is the same as that of inside, we say, this is a key word what you need to know, there is no net movement of water molecules across the plasma membrane. Why? Because equilibrium is maintained. How do I know equilibrium is maintained? You can see one water molecule entering in, one water moving out. So no net movement of water molecules in either direction because equilibrium is already maintained within the cell. We do not want to create disequilibrium in the cells. Let's go to the next scenario, hypotonic. Hypo comes from the Greek word which means less or under. So this is our red blood cell. So when the concentration of solute outside the cell is less than inside, so solute concentration, salt or sugar outside the cell is less than inside, that means there will be more water outside. So water is going to enter the cell. So if this is my red blood cell, water is entering my cell. What is happening to my cell? It's going to expand and then burst because every cell can only hold a maximum amount of liquid. The volume of a cell, every cell has its maximum volume or capacity it can hold. And so once the cell expands and it can hold no more water because it's reached its capacity, the cell is going to now burst. So the cell bursting of a cell is termed lysis. The cell is lysed. So when the concentration of solute outside the cell is less than inside, water enters the cell, causing the cell to burst or be lysed. Keyword. Let's go to the third scenario, hypertonic. Hyper comes from the word more or over. So when the concentration of solute outside the cell is more, if salt concentration is more, then water has to be less. So which, because this is osmosis, it's only the movement of water molecules into or out of the cell. So if I had more salt, I would have less salt, less water outside. Therefore, water is more inside the cell. So water has to leave the cell. So this is my cell now. Water is leaving the cell. What is happening to my cell? It's shrinking or shriveling. So when the concentration of solute outside the cell is more than inside, water leaves the cell, causing it to shrink or shrivel. Now let's see what happens. This is in our cell, animal cells. Let's see what happens in plant cells. So in an isotonic solution, what happens is The cell is said to be flaccid. Must know these terms. In a hypotonic solution, the cell is normal, turgid, firm. In a hypertonic, it's plasmolyzed. So plasmolysis occurs. And you will see this when we look at the elodially in the lab, when we do diffusion and osmosis, which will be next week. So let's look at what's osmoregulation. Osmoregulation is the control of the concentration of the solutes and the water balance, which is very essential for life to exist in different environments. Where do we see our osmoregulation? Not in ourselves, but mainly in lower organisms like the protist, as in paramecium, which is hypertonic to its pond water environment. And paramecium has this contractile vacuole, which acts as a pump to expel the water. And that's how the cells are going to maintain their water balance. So let's look at the third scenario. So we finished osmosis, we finished diffusion, facilitated diffusion. Facilitate in English means to help. So facilitated diffusion is the process where you have transport proteins that are going to facilitate or help in the passive movement of molecules across the plasma membrane. And the transport proteins would include that channel protein and the carrier proteins, which are going to facilitate the movement of ions or water molecules. Ion channels will facilitate the transport of ions. Some ion channels are called gated channels. The very word, what happens to a gate? When you enter PMCC to come for the lab class, you pass through the turnstile. Unless you tap your PMCC ID card, the turnstile does not open. So the gate opens, ions can move across the cell membrane. When the gate closes, ions cannot move across the cell membrane. So the gated channels are going to respond by opening or closing in response to a stimulus that is created. For example, in our nerve cells, the ion channels are going to open in response to any of the electrical stimulus that is being created. So we can see clearly here how solutes can move across this channel with the help of the channel proteins to the other side of the cell, from the extracellular fluid to the cytoplasm. And carrier proteins are going to carry the molecules across it. So carrier proteins also will undergo subtle changes in the shape that can translocate, that can help in the solute to bind to the site across the cell membrane. So now let's look at the second type of transport, active transport. So the opposite terms that we used in passive transport will be for active transport. Active transport is the process where substances are going to move from an area of low concentration to an area of high concentration against the gradient. So low to high is one keyword, against the gradient, with energy required for the movement. So you require ATP for the movement. So an example of active transport in animal cells is an animal cell has a much higher concentration of potassium ions and lower concentration of sodium ions compared to its surroundings. Now, this is a key pump, very important pump called the sodium potassium pump that operates in our cells. This sodium potassium pump is a transport protein that is energizing the transfer of a phosphate group by the hydrolysis of ATP. Now, what happens if this is my cell membrane? This is the inside of the cell membrane. This is the outside of the cell membrane. Now, if there is an accumulation of sodium ions on one side and potassium ions on the other side, so if sodium ions are present inside the cell here, there will be More sodium, less potassium. More potassium, less sodium. So for every 3 sodium ions that are pumped across the cell membrane, the sodium potassium pump will simultaneously pump 2 potassium ions into the cell membranes. And this is how the sodium potassium pump will create the equilibrium across the cell membrane. And so now there is something called a membrane potential. What is membrane potential? It is the voltage that is being created across a membrane. And this voltage is created because of differences that exist between the positive and the negative ions across a cell membrane. And the cytoplasmic side of the membrane is negative in charge with relative to the extracellular side. And that is exactly how the sodium potassium pump is operating with the help of ATP. So for every 3 sodium ions pumped out, simultaneously 2 potassium ions are pumped in with the help of ATP molecules and the hydrolysis. Remember, ATP gets always simplified to ADP and AMP, and that phosphate bond, when it is broken, the hydrolysis of ATP results in the transfer of organic phosphate bond being changed to inorganic phosphate, so the PI. So we saw here cytoplasmic sodium ion binds to the sodium potassium pump. The affinity of sodium for sodium ions is high when the protein has this shape. So it has to change its shape. And here, sodium binding stimulates phosphorylation. The breaking of the phosphorylation is the process of ATP being simplified, where the phosphate bond gets broken down. So it leads to a change in protein shape, reducing its affinity for sodium ion, which is then released outside the cell. And the new shape has a high affinity for potassium ions. See, the shape has changed to bring the potassium ions, which binds on the ECF side, which then triggers the release of the phosphate group. That's phosphorylation. So you can go through that. Now there is a concept called electrochemical gradient. So 2 forces are going to combine together, which are collectively called the electrochemical gradient. And this electrochemical gradient is going to drive the diffusion of ions, move ions across the cell membrane. From the very word electrochemical, it consists of an electrical force and a chemical force. The electrical force is the effect of the membrane potential on the ions movement, and the chemical force would be the ionic concentration gradient. Now, we have what we call an electrogenic pump, which is a transport pump that is going to generate a voltage across the cell membrane. One example of an electrogenic pump, very important, is the sodium potassium pump that is only operative in animal cells. Plant cells have what we call a proton pump. That is their electrogenic pump. But besides plants, fungi and bacteria also have the proton pump, which from the very word proton are hydrogen ion donors. They are actively transporting the hydrogen ions out of the cell. They are not present in animal cells. We have exclusively the sodium potassium pump. And so both these electrogenic pumps are going to help to store the energy that can be used by the cell to perform any work. Now, we talked about active transport. In active transport now, I said to you earlier, there are 2 processes, endocytosis, exocytosis. Endocytosis is of 3 types. Endocytosis is phagocytosis, pinocytosis, and receptor-mediated endocytosis. So now let's analyze the term endocytosis. Endo means within. So endocytosis is the process where the cell is going to take large macromolecules of like food molecules, or even sometimes an entire microorganism like a bacteria or virus can enter the cell. How? By forming these vesicles. So if I were to pinch my cell, like this represents my cell membrane, this is a vesicle that is formed by the cell membrane, it's sac-like structure. So that's how molecules are going to be brought into the cell via the cell membrane. Phago comes from the word phagein in Greek, which means cell eating. So phagocytosis is the process where a solid food molecule or a solid macromolecule is brought into the cell. A cell engulfs a particle in the vacuole. We see it very vividly in, or clearly in bacteria. Bacteria is going to engulf its food with the help of pseudopodia and its vacuole. It forms a vacuole, vacuole fuses once the food is trapped by the bacteria and it is or engulfed, and now it is brought into the cell. Vacuoles also fuse with lysosomes. Remember, animal cells have lysosomes. That's a typical animal cell organelle, as we learned earlier. And lysosomes are going to secrete the hydrolytic enzymes or digestive enzymes to digest the particles. So pinocytosis is another process of endocytosis. Pino comes from the word pinion, P-I-N-E-N, which means cell drinking. So where liquid molecules are brought into the cell, molecules dissolved in droplets are taken up when the extracellular fluid is gulped into, again, these small, tiny vesicles. And then we have receptor-mediated endocytosis. And receptor-mediated endocytosis is the binding of specific solutes to a receptor that is going to trigger the vesicle to be formed. So receptor proteins, receptors, and other molecules will form the extracellular fluids that are transported in the vesicles. Now, we humans, we use these receptor-mediated endocytosis to take in cholesterol, which is carried in particles called low-density lipoproteins in the LDLs. And individuals with the disease familial hypercholesterolemia, they have a missing or defective LDL receptor proteins. That's how they suffer from the disease. So coming back here, as I explained earlier, how through phagocytosis, the bacterial cell, or amoeba, in this case amoeba, will throughout its projections form pseudopodia to trap its food. Once the food molecule is engulfed with the help of pseudopodia, the vesicle closes or the food vacuole closes and the food is now trapped. Pinocytosis, where you're taking in liquids, and receptor-mediated endocytosis is when the receptors are going to bind to the molecules. So the last slide is you have glucose uptake over time in guinea pig red blood cells. It's important to go through these graphs, students. I encourage you, because graph identification is important. How to read a graph, how to interpret a graph. So here you have on the x-axis incubation time in minutes, and here you have the concentration of radioactive glucose in millimoles. And so we can see that there is clearly a 10-minute time interval in the incubation time. But what do we see? We're comparing the blue graph is indicating a 1-month-old guinea pig, and this is a 15-day-old guinea pig. And so we can see with as time increases, what we see with respect to the 1-month guinea pig, that the concentration of radioactive glucose is also increasing. But as the guinea pig is older, the concentration of the radioactive glucose is increasing at 60-minute interval. So here it is over 100, and here it is about 50 millimoles of glucose. I hope you enjoyed this recording, students. Go through it. And if you have any questions, feel free to come during office hours, ask me in the lab. Have a good one, students. Take care. Bye-bye.